I'm just highlighting a few imaging technologies. As mentioned, I was a postdoc with Enrico Gratone, who spearheaded the Laboratory for Fluorescence Dynamics. And over the years, we actually co-developed many of these techniques that I'm highlighting here. And today I'm going to talk about the phaser FLIM analysis. But just to give a shout out to FCS experiments, so fluctuation correlation spectroscopy, and thank you so much for giving me the opportunity to show during the workshop how to do RICS analysis. So this is just an example of the pair correlation function, and what you see here is a protein labeled with GFP that actually goes to the site of DNA damage. And you can look at the diffusion. Okay, so we study cell migration, and over the years, we've been interested in actually looking at how cancer cells migrate out of the tumor mass. So we know that the extracellular matrix, the alteration of this matrix can lead to invasion, survival, and proliferation of cancer cells. And specifically, increased tissue stiffness, as you heard from Maddie Parsons earlier this week, can actually lead to these changes of molecular and cell dynamics. So I'm going to highlight two areas of the work that we've done and how we actually got to mitometer and how now we're looking at the changes of metabolism as cells migrate out of the tissue. So the first work here, I'm just going to highlight this work. And before I do that, I'm going to actually introduce FLIM. So I'm sure that many of you know what FLIM is, but just to, let's say, have a brief overview of FLIM in case you have not done it or heard of it. So we're exciting the molecules that are fluorescent, and the electrons get excited. They go from the ground energy levels to the higher energy states, and eventually relax down out to the ground state with the emission of the photons. So this absorption process is very fast, in the order of femtoseconds. And when the electrons are in the excited state, this is known as the fluorescence lifetime. And the natural fluorescence lifetime is the fluorescence lifetime divided by the sum of all the possible types of rates that occur during this process. So we measure when photons arrive, and I'm not going to go into the details as to how we do this, but for the rest of the experiments that you will see, we're using multi-photon excitation and with a laser that's set to 80 megahertz. And so you can use time tagging techniques to calculate the time of arrival of the photons, and of course you can do fits. So we did not develop FLIM. What we did in this case is contributed to how we analyze the data. So I'm just going to give a shout out here to the electrical engineering field that in the late 19th century, the electrical engineering applications came up with the word phasers, and it comes from phase vectors. So essentially, you can take, for example, an alternating current here and transform this wave function into phasor space. So essentially, it becomes a geometric solution for how to analyze the data. So as you can see here, we have a vector that rotates along this axis, and the length of the vector can be defined either by the change of amplitude, and then we have this phase shift, which if you're looking at, let's say, frequency space, if there's a shift in phase, then it will give rise to the angle of the vector change. So the phasers were also developed by a few other folks, and I'm just going to highlight their work. So Andrew Clayton and Quentin Hanley developed the AB plots. That is exactly the same thing as the phasers that we know today. Bob Clegg's lab at UIUC, when I was a postdoc in Urbana-Champaign, they were developing with his student Glenn the polar plots. And these two were applied to solution measurements, and we were the first to apply it to images. So you can either take data in the time domain or frequency domain, and we convert the raw data here into G and S coordinates. This is the frequency domain, so if you're using, instead of time tagging, and you're using like modulated laser sources, you could see clearly here the change in amplitude is the modulation, and the change here from the reference signal is the change in phase. So you can eventually plot all of the fluorescence lifetime on what is known as the phasors. And if you have a single exponential decay in the frequency domain, that's when tau phase and tau modulation is equivalent. These phasors will fall on the semicircle. And as the lifetime gets shorter, it will fall on the, let's say, right side of the curve here. And just for simplicity's sake, to show you. Now, when we analyze the data, it becomes fairly easy to analyze because you have the number of pixels in your image, and all of those pixels are plotted on this phasor space. So you can essentially just take a cursor and highlight the pixels that correspond to the lifetimes that are separated on the phasor. For example, you can take a solution of NADH. This is nicotinamide adenine dinucleotide in the reduced form. It has a very short lifetime. And as you add a protein, it becomes very long to about 3.4 nanoseconds or even longer, depending on the type of protein it's bound to. And if you have a third component, you can actually calculate the fractional contributions of either two components along this axis, along this axis, or along this axis, or a fractional contribution of three potential components. They will fall somewhere inside the triangle. So the data becomes really easy, specifically if you want to do FRET. So what happens in the FRET situations, you have the lifetime of the donor, and as the lifetime starts to transfer the energy to the acceptor, then what happens is the donor lifetime gets shorter and shorter and shorter. Eventually it comes back to some sort of background. Specifically, if you're using cells, you have background signal there. And you can calculate the FRET efficiency and the linear combination of background to donor alone so that you don't make a mistake thinking that the intensity is decreasing because there's FRET. It's actually because there's more autofluorescence or background signal there. So just to give you an example of short lifetimes here, and as the lifetimes get longer, this is single exponential decays. You see that the components will fall towards the 0-0 axis. And we can calculate the fractional contribution of two possible fluorophores, depending on how much you have in solution or in your pixel. So all the fluorescence lifetime can be plotted on the phasor plot. And what's the nice part about this is a fingerprint of each individual fluorescence lifetime. But we know biology is much more complicated than this. So for the rest of the experiments, I'm going to show you just a brief highlight of how we can use the phasors. This is an embryo that's developing from the 2-cell stage. This is a mouse embryo to the late blastocyst stage. And what's interesting about the mouse embryo is that the lifetime actually has a switch every time it divides. So from that 2-cell to the 8-cell to marula stage, there is a unique fingerprint on the phasor plot that you could see on the phasors. Now, if you cannot see it, all you have to do is take your cursor here and highlight the pixels that belong to the contribution of this lifetime, which we see in this 2-cell phase. And as it goes to the 8-cell phase, the lifetime gets shorter. And as it continuously develops, the lifetime gets much longer. And it goes towards this particular part, which is long lifetime species. And I don't have time to explain why that happens, but we have found out why this happens. It's due to the production of reactive oxygen species. And that's giving rise to oxidized lipid signatures. So how do we know this is true? Well, if you go back in the 1950s and read this really old data, you could see that the 2-cell, 4-cell to 8-cell and blastocyst stage actually have unique lifetime, unique metabolic switches. So this was all done crushing the embryo and essentially expiring the embryos, but our lifetime measurements were done just with the free component or bound component of NADH. And so what we see here is that the free lifetime, the free NADH fractional contribution should go down due to pyruvate uptake. So we see a shortening of the lifetime. And the free NADH will go up, so the lifetime gets longer, and we have those switches, as you see in that region. So the lifetime goes from long to short to long. And what's really nice about this is that the phasor FLIM can measure metabolic during embryo development in pre-implantation embryos. And so you can start to understand that this could be a very valuable resource for IVF clinics. Okay, so now back to our example of the cell migration. So what we know from the literature is that if you increase the rigidity of the matrix, this is an example that was published in 2011 from PAA, which is this substrate, polyacrylamide substrate, and as it gets stiffer, the invatopodia actually start to increase activity. So what's important to note, and I definitely don't want anyone to get kind of confused about this, is that invatopodia are invasive tools. They try to degrade the matrix so that the cells can migrate. But the leader cells and the cells that we're interested in looking at are the cell protrusions. So as the cells start to invade the tissue, we're looking at those navigational tools because those are the mechanotransduction properties that the cell needs to migrate forward. That's where focal adhesions appear. And then, of course, either the lamella or the filopodia can explore the microenvironment and react to extracellular cues. So metabolism also modulates cancer cell invasion. This is an example that if you did degradation of the matrix, as you switch their nutrients, the degradation increases. So this is also giving us a hint that metabolism is playing a very important role in cancer cell invasion. So to look at let's say, metabolic imaging of these particular cells, we look at nicotinamide adenine dinucleotide. And this is a really interesting molecule because this molecule has a very short lifetime when it's free. So what you see is the nicotinamide and the adenine group overlap and it's essentially quenched. So the intensity is very low and the fluorescence lifetime is short. When an enzyme binds to the reduced form of NADH, this opens up and the lifetime gets longer and the fluorescence intensity is quite bright. So I'm not going to go into all the details, but just suffice to say that NAD plus is not fluorescent and it's very hard to distinguish the lifetime of NADH and NADPH. So in case you're wondering about that in the audience, the higher, let's say, fraction that is made by the cell is NADH, not NADPH. So, okay, so a lot of other groups have used the free and bound fraction of NADH as a metabolic marker. And Melissa Scala's group has looked at the in vivo multiphoton fluorescence lifetime imaging and protein bound in precancerous epithelia. And Chiara Stringari has also shown that the phaser film of free and bound reveals, let's say, stem cell differentiation and potential. And what these papers are showing is that if you have a higher population of free NADH, your cells are actually undergoing more glycolysis. If you have a higher population of the bound NADH, the cells actually are undergoing a process called oxidative phosphorylation, which is what is found in the mitochondria. Okay, so you can take a spheroid. These are cancer spheroids, and these are, let's say, non-tumor cells. These are fibroblast cells that surround the spheroid. You can perform the FLIM analysis with the phasers and map out the fractional contribution of free and bound NADH. And what you see in this particular example is that the fibroblast cells have a very different metabolism than the tumor itself. So, and this is in color-coded to show the higher bound population of NADH and the free population here in the pseudo-color map. So what we're interested in is how the ECM impacts cellular metabolism. And for us to do that, we took collagen, and first we actually calculated the density of the material, of the collagen fibers. And to do that, we perform image correlation spectroscopy and obtain the spatial correlation. Of course, these are not moving, so they're fixed structures. So what we're actually correlating is the size of the structure in our image. And this was using second harmonic imaging. And the standard deviation gives us the actual average size of the structure, and the amplitude function is a signal to background, which is proportional to the structure. So under two different ECM densities, 3 megs per ml of collagen or 1.2 megs per ml of collagen, you can just visually see that this is more dense than this structures here. And we can calculate the fiber thickness. There is a statistical difference between this and then the density based on the amplitude of the correlation function. But if that's not enough, we also use rheology measurements to measure the modulus of the ECM density. And you could see that they are statistically significant. All right, so I'm just going to summarize what we found. What we found is that MCF10As, these are normal fibroblast cells. And under the stiffest material that we have, which is glass, you see that the metabolic index here is more oxphos. So these cells don't change whether we plate them on 3 megs per ml or 1.2 megs per ml. We also use a ROCK inhibitor to show that contractility may or may not impact the metabolism. In the case of non-cancer cells, nothing happens. For PR positive cells, also the same thing. Not too much change in cellular metabolism, but surprisingly, for triple negative breast cancer cells, they do respond to the ECM density. So triple negative breast cancer cells, we see that there is a change in free and bound NADH. And if you knock down the ROCK, let's say you use the ROCK inhibitor, you see that now on glass, the metabolism has changed. So we can manipulate the metabolism, the triple negative breast cancer cells, showing that they're still pliable. So overall, what we see is that collagen, the soft matrices increases oxidative phosphorylation. And this is really important because in a collaboration with Stephanie Sillett-Slab, who's now at UT Austin, and her postdoc, Ali, they use glioblastoma cells, and they use a material, hyaluronic acid, and they can tune the density of the material and the stiffness of the material. So these are cancer spheroids, glioblastoma multiforme spheroids, on stiff condition and soft condition, and they image them after many days. So in the first day, the tumor pretty much stays circular. There are no cells that are actually migrating out of the spheroid. In day 3, we start to see a few cells appear in the soft condition. And the soft condition, you see these cell protrusions after 6 days. So Stephanie's lab does bulk RNA-seq measurements. And what you see here is 2 different patient-derived glioblastoma cell types, the HK408 and the HK177 multiforme. And what they see is that the stiff material here is upregulating and these genes here is really, really hard to see, the mitochondria genes, but downregulating in the soft material. And the same thing happens with the H177 cells. So that was a little disappointing because we thought that, oh, the cells that are in the soft, right, would be more oxphos, but actually they're not. These are more, they have the opposite of what we expected. So we did here the FLIM in this, the spheroids, these 2 cell types, and in the stiff and the soft material, and you see the fraction of the bound NADH. So in the bound, so this is more blue, this is the fraction of bound NADH is much lower, but in the stiff material, the fraction of bound is much more increased. So at the end of the day, it seemed like soft matrices are more glycolytic. However, my students said, let's take a look at the cells that are migrating out. So instead of just looking at the tumor itself, let's look at the cells that are invading the tissue. And what we found is that these cells that are in the soft material increase their cellular metabolism to be in the more oxphos state. So while the tumor itself is more glycolytic, the cells that are migrating out have a different metabolism. So you would completely miss that information using bulk RNA-seq data and if you're using another, let's say, secondary method to look at cellular metabolism, which could be seahorse assays. So in another collaboration with Devin Lawson's group, she does these xenograft models where they take triple-negative breast cancer cells from patients, put them into the mice, allow those cells to migrate into different parts of the tissue, including the lungs, the lymph nodes, and the tumors. They isolate these organs. They dissociate the cells, and they isolate the cells that migrated in these regions. And they know which exactly are the cells because these are human cells that are expressing CD268 or 298. They also perform single-cell RNA-seq, and they wanted to know what genes are upregulating metabolism. So the micrometastatic cells, the ones that went to the organs, are upregulating OXPHOS, and the cells that are at the tumor that did not migrate are upregulating canonical glycolysis. So that also matches what we saw. And what's interesting is that if we use an inhibitor to knock down the OXPHOS, so let's imagine this. Let's try to not get these cells to migrate. What's the best way to do it? We thought, okay, they're highly glycolytic. Let's shut down their OXPHOS pathway so they cannot switch to OXPHOS. So by doing this, we use oligomycin that shuts down complex 5 in the mitochondria. And we verified the analysis with FLIM, and we see that 0 hours post-treatment, this is the controls, and then after 24 hours, we see that there is a switch after treating the cells. So for sure, by using the oligomycin, we're actually increasing the fraction of free NADH, making them highly, highly glycolytic and shutting down the OXPHOS pathway. And we did that for these MB231 cells, triple negative, and the other triple negative cell types. Then they put these cells into the mice after shutting down the OXPHOS pathway, and they counted the number of cells that went to these organs. And here's the data over here, and you could see that the oligo-treated cells were much, much less than the cells that were not treated and put back into the mice. So at the end, you could see that both the glioblastoma and the breast cancer cells at the primary site, they're highly glycolytic. Under the soft hyaluronic acid, they're highly glycolytic in the core, but the metastatic cells are upregulating OXPHOS. So that means that the cells have a switch to increase their invasive potential. So we were much interested in how that happens, and we know that mitochondria is key. So my student looked into some papers, and he found that mitochondria are actually, the phenotype, the size of the mitochondria, can actually give you a lot of information about the cells. So these are not our papers, these are cells that differentiate into glial cells, and you could see that the intermediate, let's say the long mitochondria, which are these gray pixels here, are differentiate into glial cells. The intermediate size mitochondria differentiate to progenitor cells, and the short mitochondria differentiate into neurons. So mitochondria is doing a lot of different things in the cell to regulate cell function. And there is another paper that was seminal that talked about this protein called synaptophyllin. And what's interesting about synaptophyllin is that it's a stop molecule. So we reasoned that mitochondria is providing energy in regions of the cell, like cell protrusions, that need ATP to migrate out. So we found that if synaptophyllin is responsible to stop the recruitment of mitochondria to the leading edge. So if they use sRNA to shut down the production of synaptophyllin, what happens is that the recruitment increases the transport of the mitochondria, but not just that, there's no longer a stop sign. The mitochondria are free to actually go to the regions to provide ATP. And the cells, they become more invasive. They travel faster and they're more metastatic. So I wanted my student to follow these mitochondria, but the cells take a long time to move. So we said, okay, let's do something very quick to see if we can get the cell protrusion to be led. And one way to do that is to use photoactivatable proteins. This is RAC, that is a GTP protein that in the GTP or GDP bound state, it enables the cell to migrate in the direction that you shine the laser onto it. And the way that this works is this is a love domain. This is a photosensitive protein, and as you add light to the system, it undergoes a reaction that transforms this to a different structure and that releases, so RAC becomes activated, and that's what causes the cell to move. So here are photoactivatable RAC, let's say directed motion of mitochondria. So this is where we are photoactivating RAC, the protrusion increases, and we see the, let's say, the transport of mitochondria towards that leading side. This is in triple negative breast cancer cells. In let's say MCF10A fibroblast cells, we do not see this, let's say, preferential transport. My student here moved the laser so that you could see that even in this part, there is no preferential region of mitochondria tracking. So we did many of these studies, and what we found is that if we photoactivate RAC, there is a directionality preference. If we don't photoactivate RAC and just wait for a long time for the cells to move, we do see still a directionality ratio increase. But for the control cells, these are non-cancer cells, there's a decrease in the directionality ratio. And of course, the speed of the mitochondria also increases as we photoactivate RAC. And so you might ask, how does this work in collagen and cells that are, let's say, have these leading edges? And we labeled here the mitochondria with COX-8-GFP, and you could see that the cell is protruding here. And when we refocus this, because this is in 3 dimensions, you see the mitochondria are at the leading edge in both cells, whether they're in 3 megs or 2 megs per ml. We also use FLIM to measure the free and bound ratio of NADH in this protrusion, and from 0 minutes to 15 minutes, we do see that there's a free NADH production in these regions of the protrusions. Now, this is ongoing work, so we don't have any direct conclusions from the free and bound NADH ratios of the cell protrusion. But we are interested in the mitochondria structure and phenotype and branching and other dynamic processes that happen. And my student at this point was tasked to track mitochondria over time. And the best way to track this mitochondria is to use, let's say these papers use ImageJ or Fiji, and these are manually tracked, right? And when you look at mitochondria in this particular image, there's a lot of background signal, and it's very hard to identify one mitochondria from the other. So taking this data is easy, but analyzing the data is hard. So if you do 100 of these mitochondria and you click each position for each mitochondria, it's going to take about 10,000 clicks just for one condition. And my student came to me and said, there is no way I'm going to graduate if I keep doing this analysis. So we said, okay, so and it's also bias particular to the operator. All right, so we came up with a solution. We decided to automate this process. So here's the original image, and yes, you can take a picture of this and go to the mitometer, which is the algorithm used to identify each individual mitochondria, track them, and get all the information that you want. So you can see there's a lot of background in this image. So we borrowed from astrophysics, and we used the diffuse background subtraction algorithm. That's what the diffuse background looks like. If we subtract that diffuse background, this is what we're left with. We also Gaussian filtered, we thresholded it, and then we have a very clean, identifiable mitochondria for this particular cell. We also automated the process for tracking the mitochondria in terms of looking at the mean square displacement and all of the other functions undergoing fusion and fission events. And this is just an iteration to show you that we can use medium or different types of diffuse filters. This is here in this particular region. If you zoom into these two boxes, you'll see that this is really, really blurry. So this is the diffuse background subtraction on high condition. And then we are able to identify the underlying structure in this very, very bad background. And if you have very low background, we use a low background subtraction here, and you can identify that there is an actual structure here, underlying structure, that is identifiable. So we are able to quantify many of these kinds of structures within the same image. So I don't have time to go through all the details, but I'm happy to share if you're interested during dinner or via email. We can calculate the fission and fission events and make this automated. So we have an algorithm that looks at each individual frames. So if you have one mitochondrial structure, and then in the second frame, it turns into two objects, we keep doing this iteration so that it identifies this kind of fission event. Of course, if you have two structures and it turns into one structure, there would be a fusion event. So we can calculate the fusion and fission events. Because we're able to visualize, we're able to track the individual mitochondria and obtain the mean square displacement and thereby get the velocity. But we can also multiplex this technology by using the NADH autofluorescence signal. So even though we are, let's say, tracking the mitochondria with a fluorescent dye, we can use NADH. NADH emits in the blue region of the spectra, so as long as it's spectrally separated, we can simultaneously excite NADH and the other fluorophore at the same time and get two different channels. And in the lifetime data, we're able to obtain, let's say, the free and bound fraction of NADH. The problem with FLIM is that the mitochondria move, so you're going to get a streak pattern, and this is not what we're tracking. We're tracking the other channel, but we're still obtaining the lifetime of NADH. So if the mitochondria move, what happens is that we continue to track these mitochondria, and we just get a lifetime, let's say average, of each position to obtain a final fluorescence NADH lifetime for the mitochondria. All right, so what does this give us? So here's the mitochondria that has been, the mitometer program has identified. We use MCF10A cells, non-cancer cells, then cancerous but not as aggressive cells, and these are the triple-negative breast cancer cells. And what we see is, first of all, the number of mitochondria is different. The triple-negative breast cancer cells have a large number of mitochondria compared to the controls, and even more so than the ER/PR positive cells. And the fractional NADH contribution within each mitochondria, and you can see this, these are all color-coded to calculate fractional bound contributions within the mitochondria, are heterogeneous. So they don't all have the same, let's say, NADH population. And you can identify which mitochondria have more, let's say, less bound NADH than the other. And you can see the triple-negative breast cancer cells statistically do have a higher fractional of population of bound NADH. So you can use all this data set and look at morphological and motility parameters in respect to bound NADH to see which of the features that are more of interest for the triple-negative breast cancer cells. Okay, so you might be asking, this is all done in 2D, can we do this in 3 dimensions? And the answer is yes. So we took these tumor spheroids, and these are patient-derived normal epithelia. These are patients that have breast reduction surgery from our clinic, and we can culture these. And we also took ER PR positive cancer cells and the triple negative breast cancer cells and grow them in three-dimensional spheroids. So we compared the differences between 3D and 2D, and this is a lot of data, so I'm gonna go quickly as to what the differences are. So in 3D cultures versus 2D cultures, we see mitochondria are slower, they're more directional. They are less burst of speed, and let's say the matches cell migration in 2D versus 3D, we see that the triple negative breast cancer cells undergo more fusion events. That means that they actually have higher invasiveness. The triple negative versus ER-PR positive cells have more solidity, and I didn't talk about this. This is the branching. So if you get a solidity value of 1, that means it's a complete solid structure. But if the fraction is lower, then that means they're more branched. And then we have lower membrane potential because you could use a functional dye to calculate the membrane potential and actually look at the phenotype, and they're less rounded. So in general, normal cells look very similar to the breast cancer cells, but very different than the less aggressive tumor cells. And you have a lot of data. So all this data can be put in a random forest classification system, and you can use different, let's say, train sets, and by looking at the motility features, both combined or morphology features, to calculate the feature importance. And that's important if you're trying to identify cancer cells that are perhaps more chemo-resistant than non-chemo-resistant. And you could see that in all 3 cases, for our the motility features, morphology, or whether they combine, we see better than random guessing predictions. So it's a very good tool to look at maybe cancer cells as they're being treated with chemotherapy drugs. So this is what some of the mitochondria look like in 3-dimensional space. This is the hyaluronic acid soft and stiff that you saw earlier. These are the glioblastoma multiforme cells in 3D. And we're able to look at the soft matrices versus stiff matrices, and we see that in the soft matrices, they traveled further, they're more directional, they're less branched, and they're longer. So as you recall, the soft matrices had more glycolytic for the tumor itself, and then the stiff hyaluronic acid matrices have more oxphos increase. Okay, so I just want to wrap up here and show you that Mitometer is a really useful tool to be able to track mitochondria, and depending on your application, you can actually use it for many, many different things, but you can also multiplex this with fluorescence lifetime imaging to get the data. And for this part of the work, I'd just like to thank Austin Lefebvre, who's now at Calico, and my former postdoc and senior researcher Lorenzo, who's in Toulouse here in France, and all of the folks and my collaborators in funding for this project. But I also wanted to share that if you're interested in looking at the phasers, we do have, we developed a Python library with Lionel Malacrida. So you can actually go to the phaser pi analysis. And I just also want to invite you to come to Irvine, California, which is 10 minutes away from the beach. So we're near the beach cities like Newport Beach, Laguna Beach, Huntington Beach. So we're literally next to Newport Beach. And this year we've actually partnered with UC Berkeley's AIM workshop. So perhaps you may have heard of it. Holly Aaron runs a workshop up in Berkeley in January. And we're partnering together with UC Berkeley to do the workshop this year. And we're going to do a lot of this kind of analysis and feature many of the vendor that you saw here as well come and bring their instruments. Okay, and that's the last thing I want to lead with, and I think I'm way over time.
No, it's okay, 5 minutes.
Okay, is can we use the phasers for another type of analysis? And you can for hyperspectral imaging. So in this case, you can collect, let's say, 32 channels with a commercial microscope and identify the spectral emission depending on, in this case, hyperspectral data. Transform the spectral data, as you could see here, the center of the emission signal into phaser space. So as this emission broadens, the phaser gets closer and closer to the center axis here. This is just a pictorial demonstration. And of course, if you have multiple components, you can calculate the fraction of one to another. So what's really nice about this, and we were not the first to actually come out with the spectral phaser. Farzad Feridorni, as well as Hans Gerritsen and Arjun Barter, came up with this transformation. And you can calculate the fractional contributions of these spectra using the phasers. But what is maybe more important, I think, is let's say that you go to your microscope and you don't have a spectral detector. So what do, right? Some of these things are very, very expensive. So Enrico came up with a very, very nice solution, very cheap solution. In fact, it's so cheap that the first sine and cosine filters that this was Sasha's work, that they purchased was from Amazon for $5. So they bought these filters from Amazon for $5. So it's really cheap cosine and sine filters. And essentially what they did is they put it on the diver. So the diver is a deep imaging microscope developed by Enrico and Sasha. And what they saw is that if you use, let's say, a sine and cosine filter, you get this kind of overlap spectra. And if you put them together, they should make a perfect circle. But because these were super cheap filters, they are not. So they had to do a correction in radians for each one of these, let's say, wavelengths of emission. So these filters enable you to actually detect the emission directly, and calculating this into phaser space to be able to obtain the spectral separation of your fluorophores. And I'm not going to go into all the details. There's a lot of details here, but they use a monochromator here to measure the phaser wavelength emission, and you could see the separation of each one of these specific wavelength of emissions. You can also calculate the fractional contribution of two components. This is pure coumarin mixed with rhodamine 6G. At 50-50, they're purely halfway between the two axes. So we use the sine and cosine filter for a bioluminescence imaging system that we engineered. And these sine and cosine filters enables us to collect the, let's say, the first wavelengths here from 400 to about 550 nanometers and beyond for the cosine filter. And what's nice about this system is that you don't need any lasers. So you're just basically need a camera to identify the intensity at each one of the pixels. And because we have two cameras, we get the, in this case, the entire spectra. So this was the first time that bioluminescence community can actually identify spectra using a microscope that is not commercial. There's only, I think, only one commercial microscope that is available to them. So, and please correct me if I'm wrong about that. If there are more, let me know. So these are all the reporters by my very wonderful collaborator here, Jennifer Prescher, and her two students, and then Lorenzo, who helped to engineer the system. And what you see is the spectra of each one of these reporters separated on the phaser plot. Then we mix the cells together, and you could see that they are highly separated. Of course, they're spatially separated because they're only expressing one of the many reporters, but we can identify them all at the same time in the same culture dish. So you can also do tracking of the cells, longitudinal tracking. These are two reporters, a nuclear reporter and a cytoplasmic reporter. And what's nice about this is that you can measure this over a very long period of time and separate out those emission spectra. And if you're in the bioluminescence community, we can also calculate the Brett efficiency of the reporters from the donor to acceptor based on the fractional contribution. And so we showed proof of concept here with the Brett reporter and different Brett efficiencies for the sine and cosine filter. So 2 cameras was a little cumbersome, so now we've engineered a system where we only have 1 camera. So this camera is split into 3 channels, a reference channel, and a sine and cosine filter here. So if you're interested, you can purchase now much better sine and cosine filters from a company called Ausora. And this is custom-made filters. All right, so I'm just going to stop here and thank you all for your attention.
I have a question regarding the NADH bound and free NADH. You talk about the reactive oxygen species in the cell, and we know that regarding the microenvironment of the fluorophore, the lifetime will change. And I was thinking about the reactive oxygen species interacting with the NADH, and how can you see it on the phasor, because you don't know if you see the free or the bound NADH, or if it's only the reactive oxygen species interacting with the fluorophore on the NADH, the free or the
Excellent
bound one.
question. And one thing I didn't mention is that the reactive oxygen species is interacting with lipids. So it's oxidized lipid signatures that we're actually obtaining. And this was confirmed by one of Enrico's former students, Rup Sadatta, where they looked at the accumulation of adipose tissue, and they fed fatty acids to the tissue. And they saw the production, first the reactive oxygen species occurred, and then the oxidized lipid signature increase, and that was confirmed by CARS imaging. So what is really happening is that the reactive oxygen species is changing the lipid states, and that's the signature that is coming up. In the embryos, this happens, it has to happen. Reactive oxygen species is a normal production during development. And so we confirmed that this was due to oxidized lipids using third harmonic imaging and looking at CARS imaging.
Okay, thank you.
Yeah.
Do you have? Okay.
Thanks for the great talk. I'm sorry for the extremely naive question, but I don't know what is a sine or a cosine filter.
Yes, so this is special filters that transmit light from let's say, have a certain frequency of wavelength. So from 400 nanometers up to about 550 for one of the filters, and then starting the cosine filter from 500 to about 800 nanometers, so 750 nanometers.
So it's a bandpass filter, or
it is a type of filter that is like a, I can talk to you about that. It's
Okay.
a specially custom-made filter that has thin coatings on it. Yeah.
Thanks.
Yeah.
My question was sort of related to the first one, which is, what about other biomolecules? So like FMN is pretty fluorescent. Does it have a distinctive lifetime?
Yes, it does. Angelica Ruick from ULM, from Ulm, does a FMN imaging. And as far as I know, the lifetime is different, and the excitation is different as well. So you can use FMN as one of the markers for cancer cell states.
And what happens if the lifetime's not mono-exponential, as many are not?
Yeah. So those lifetimes, as long as you fingerprint what you can identify that particular lifetime with that excitation, right? So we use a specific excitation. If you want to excite FAD, you look at the NADH FAD ratio. You have to. So there are different wavelengths for different fluorophores of interest, and there are many fluorophores that you can look at. We only looked at NADH in those examples. Yeah.
Does anyone have another question? Okay.
Thank you, Michel. I have a question regarding the fact that you can combine the two phasor, the wavelength phasor and the lifetime phasor.
Yes.
Is it possible?
Yeah, other people have done that. So Lorenzo and Enrico published a paper where you can simultaneously collect the spectral and lifetime phasors. So yes, and there's actually a commercial company that produces the SLIM, which is spectral and lifetime.
But and also with this kind of phasor, because it's really impressive, this circle, no? Okay.
Yeah, I think that so Lorenzo's paper was simultaneous, but you get the data in different matrices, right? So you have your spectral detectors, right, for this yeah, I think the commercial microscope has integrated both. So you get spectrally resolved lifetime data. I don't know if they use the phaser, so I don't, yeah.